There. This is going to be part 2 of our chapter 10 discussion on agglutination and precipitation reactions. We just ended the chapter discussion part 1, looking at the precipitation curve, the prozone, postzone, and zone of equivalence. And we were moving into specific laboratory techniques to measure antibody-antigen binding and the formation of antigen-antibody complexes. There are a number of precipitation reactions we're going to be looking at: turbidimetric, nephelometric, radial immunodiffusion, Octolani, double diffusion, immunoelectrophoresis, and immunofixation electrophoresis. This table is a nice, really quick overview of what we're doing. And if you look here, it tells us the principle of each test. So that's a helpful thing for you to look at. The point of measuring precipitation reaction is we're going to be looking to see what the level of antibody-antigen binding is happening in a patient sample. We can do this, as I said, by a number of different techniques. The first two are measurement of precipitation reactions by light scattering. So if we remember back, when we talk about precipitation, we're talking about an event where a soluble antibody binds to its specific soluble antigen, and they form an immune complex, an antibody-antigen immune complex that is now insoluble. It forms lattice formations and it falls out of solution. It's going to be visible to us. And so when the antibody-antigen complexes form at a very high rate in our test method, the precipitate falls out of solution. And we get that, kind of a cloudy or turbid appearance. I don't have a picture exactly of it on the next slide, but we'll see. Turbidimetry measures the turbidity or cloudiness of a solution by measuring the amount of light that directly passes through the solution. That's an important fact, and I'm going to show you this in just a second in the test methodology. Nephelometry, on the other hand, is an indirect measurement. It measures not the amount of turbidity by passing the light through the solution, but rather it's measuring the light that gets scattered when light hits the antigen-antibody complexes. If you think back to our discussion about flow cytometry, we talked about how a laser, when it hits the cell, light scatters. That principle is basically the same idea in nephelometry, where we're measuring the amount of light that gets scattered when it hits the antigen-antibody complexes. So let's look at one more slide about precipitation, then we'll go into the techniques. Precipitation is the immune complexes that form once we've had that lattice formation happen. Remember, the lattice formation was one immunoglobulin bound to epitopes on two different particles, whether it's a cell or something else, and that we end up with that linking of many antibodies bound to two particles, and you end up with that cross-linking, and it becomes visible because it gets very heavy and it falls out of solution, becomes insoluble. So this picture is illustrating to us that the immune complexes is formed, and they got quite large, and they were so heavy they fell out of solution. We call that the precipitant. This is the complexes that fell out of solution. The tube on the left is obviously indicative of the formation of immune complexes, antibody-antigen immune complexes. The tube on the right, there's no precipitant here. The tube on the right has no reaction, so no antibody-antigen complexes were formed in our test method. Both turbidimetry and nephelometry will measure the cloudiness of these solutions. To test it, we would have to reinvert these and shake this up. We're basically going to measure how cloudy it is, and it's directly proportional. So if when we're looking at turbidimetry. If there's a lot of cloudy solution, very little light shines through. And when we're looking at nephelometry, a lot of immune complexes will scatter a lot of light. So if you think about it, sort of like, again, using an analogy of a flashlight, if you're shining the light through a very gauzy and kind of light curtain, a lot of light passes through it, right? We would assume that could be analogous to just a little haze or a slightly turbid solution. And we can compare that to shining a light towards a very heavy curtain, and very little light passes through that. And that would be analogous to a turbidimetric measurement where there was a great deal of immune complexes formed so that light didn't pass through. So let's look at the turbidimetric test methodology. What we're doing is we're basically, let's look at the illustration, and I think it'll make sense. We have a light source here, and we have a lens that's going to, sort of focus it in a direct way. We have a test well or a cuvette, could be a test tube of some kind, where we have a well-mixed sample, the liquid that's being measured, and we're going to see how many, what's the level of immune complexes here. We shine the light directly through the cuvette, through the glass cuvette. As the light hits it, some of it goes off to the side. Right now, we're not measuring that. We're measuring the light that passes through this solution, and we can measure it by optics on the other side. That then gets, right here is the optics, gets amplified, very similar to the concept of what we talked about in flow cytometry. The signal gets amplified and gets sent off to a computer, or to even depends on the analyzer. If it's a simple analyzer, it's just going to give us a reading of how much light passed through. The more antigen and antibody binding that takes place, the more immune complexes are, the cloudier this solution is. The cloudier the solution is, more light hits more of these immune complexes and scatters off to the side, so less goes through. It's directly proportional. Is it directly? No. It's a high degree of immune complexes means very little light gets through. A low degree of immune complexes means a lot of light gets through, so it's inversely proportional. Couple other things about it. So again, that's just the definition. Remember, soluble antigen, soluble antibody, joined together, they end up being an insoluble immune complex that precipitates out. More clouded the solution, the less light that passes through. Lower light passing through means there's a higher concentration of immune complexes. So what do we do with this information? We know how much light passes through. That doesn't really help us to quantitate what it is we're looking for. To determine the amount of substance that we're testing for, we're going to compare the light passage through our patient sample, and we're going to compare it on something called a standard curve that's prepared from known standards. Let me just go to the next slide and you can see it. I'm also going to show you a quick video about this. What we do when we prepare a standard curve is we buy calibrators that have a known value. And it's for whatever it is we're testing for, whatever the test methodology is. We use standard curves in my old lab for a backup confirmation method for hemoglobins, for example. You can do it for all different things, but in this case, we're looking for the concentration of either antibody or antigen. So for standard curve, you have to always have a minimum of 3 calibrators. 5 is better, but you have to have a minimum number. They usually come in 3 levels in a box, and if you need to make lower concentration, then you do you dilute it. You dilute your calibrators, and you make some lower concentration standards. So we buy these 3 levels of calibrator, and then we're going to run them through this test methodology this, we're going to do the test just with that, and we're going to measure the optical density that comes out on the other side. So for example, level 1 had a calibrated, a known, this is tested at the manufacturer, they know for certain that there's 5 millimoles of whatever it is we're looking for in calibrator 1. So we're going to plot the millimoles that the manufacturer tells us and the optical density reading, the light that passes through, and we capture that information, the optical density of each of these standards. So at 5 millimoles, we put a dot, and then we read the optical density, and we got an optical density of 2. So we're going to plot this on the graph. So we find the point where the optical density 2, we draw it over, and I'm not a good drawer with a cursor, hits 5 millimoles. And where they hit, you put a dot. Then we do level 2, which is 3 millimoles and an optical density of 1. So we go to optical density of 1, draw it across until we get to 3, and we can see we're right at 3. Bam, we put another dot. And then we do it again. 1 millimole has an optical density of 5. So we go to 0.5, optical density of where they intersect, you put a dot. Boom. We could stop there. We're going to draw the best line we can between using these 3 dots. We used this dot, and this dot. You see it's not a perfect straight line, and that's okay. You usually want to draw what's called the best fit line. One's right almost on this one's right on the line. It goes through the 0 axis, because if we have no concentration of solution, there should be no optical density, right? So we're looking at our highest point and drawing it down and seeing we're going to use this as a point, this is a point, this is a point, and this is a point. So you can see this is a little bit below. We also ran a couple of others. We made some dilutions off of this 5 or the 3 for this one. And we see this is slightly above the line. This is slightly below the line. This is just about perfect on the line. This goes straight through the 0. And this is pretty close. So we're going to draw the best fit we can so that it's sort of hitting right in between, a little bit above, a little bit below, and as many points as possible on the line. So we call that the best fit line. Then we're going to go read the patient. And so I gave you an example. The patient has an optical density of 1.5. We don't know what the concentration is yet because all we did was this. We read the cuvette, we put the patient test in, we shined the light through it, we got the reading through the optical element, and we got an optical density reading of 1.5. So I'm going to go up to 1.5. I don't need a second point now because I'm going to read this against my known standard curve. I'm going to take it, go over to 1.5, draw it over the best as I can here, and it is, let's see, well, that's clearly not true. Patient optical density is 1.5. If I look at this graph, it sure doesn't look like 3.2 millimoles, but might be a typo. Let's look at it ourselves. At 1.5, draw it over to the standard curve, I would get 4.2, not 3.2. Must be a typo. We're going to call that 4.2. I'm sure anyone that's ever looked at that before was like, Andrea, what are you doing? Let's see. So 4.2. So we're going to draw a line across and measure down, and that's our optical density. That's our optical density. Draw it over to the standard curve, draw it down, roughly 4.2. We would also run quality control. So quality control, unlike these standards, the standards we know what the result is exactly. Standard one is exactly 5 millimoles. Quality control is just, I don't have it here. Quality control range. Quality control gives us a range. So we again will have 3 levels of quality control. There'll be a low, a normal, and a high. And we know that the high is assayed at a particular range. So, for example, quality controls have a range. It's, that quality control level 1 is going to come somewhere between 43 and 56 milligrams per deciliter. It's not exact. The calibrator would say 50 milligrams per deciliter is what this is. Controls are a range, and when you run your controls against a standard curve, they should come within the reported range that was set by your lab. So there's a video here. I don't want to run it right now. I would like you to come back to your come back to the video if you have any questions. Actually, let's look at it really quick. I think we do have time. Let me see if this is going to work.
So we finally come to the penultimate moment in our exercise. This is where we wanted to be.
Okay, that is not. I'm not hearing that. And that, frankly, is not the one I wanted to look at again anyway. I actually changed that out. I'm going to email you the link for this. I'm going to make myself a note to send you the link. Send the link for the new video. It's much shorter. It's not 12 minutes. It's like 3 or 4. And so we'll take a look at that. Please go back and look at it. It's she'll actually draw it out and see. If you haven't, you may have done a standard curve before in intro class, and you may already know how to do this. But I'd like to make sure you need to know how to do this. So standards are the known, they're a calibrator. Quality control has a range. It's telling us what it's helping us to interpret the standard curve to make sure we drew it correctly. Turbidimetric testing is very simple, light passing it through, but it's not that sensitive. It's not a great test method because it's subject to a lot of error. Nephelometry uses a very similar process. We use this in clinical laboratories because it's pretty easy to automate. It's based on the principle that a dilute solution with particles will scatter light at, usually, roughly a 70-degree angle so this would be a 90-degree angle, right? If it's passing through here. So it's about a 75-degree angle. The amount of scatter gets collected and then amplified and sent off to be reported. So it's the same concept, but instead of looking at the light that passes through, we're going to look at the side scatter for this. We're going to look at the scattered light. Nephelometry, the good news is it can detect either antigen or antibody, but it's usually run with a known antibody as the reagent and the patient antigen as an unknown. So if we want to see if a patient has an antigen, let's use a simple one. We want to see if the patient has a certain strain of flu. They don't have to do this anymore, but I'm using this as an example. They want to see if a patient has a certain flu. They can purchase the antibody to the exact form of flu that we're looking for, and you mix the reagent antibody plus the patient's serum containing the antigen and see what happens. See if they form immune complexes. There's a couple of different kinds of nephelometric tests that can be done. The first kind of nephelometry tests are called endpoint tests. In this case, what we do is we mix the reagent antibody plus the patient antigen, and then you wait until all of the immune complexes form, and you're going to be monitoring that. The instrumentation will monitor it and see when there's not an increasing in the cloudiness. Problems with this, you don't know when it's going to end. So the timing, it can be quite time consuming in the lab, so that's not a great thing. The longer that it sits there while you're waiting for that endpoint to come, the complexes can get very, very large. Those lattice formations can get very large. They fall heavily out of solution and they become difficult to measure. If they are really big and they fall out of solution very quickly, when you go to put them in the test method, and this stuff is falling out really quickly, you're going to end up with false negative results, falsely decreased results, because all of the large immune complexes are on the bottom of the tube. There's another kind of nephelometric tests called kinetic testing. And in this case, we just add the patient antigen plus the reagent antibody, and then wait a certain measured amount of time. The manufacturer tells us you need to wait 15 minutes, 30 minutes, an hour, whatever that specific test method tells you to do. The manufacturer can tell you what the expected rate of formation should be, so many millimoles per hour or whatever it is we're measuring. But we're still going to use a standard curve, looking at the light scatter to be able to tell what the patient antigen is. Remember, if you don't form any complexes, it means the antibody-antigen reaction didn't take place. It means the patient didn't have the antigen to the reagent antibody. Those are our light techniques. So let's look at the next technique, which is passive immunodiffusion. And if we look at our table, passive immunodiffusion are a number of techniques where we're going to put antigens and antibodies into an agar gel. They migrate towards each other within the gel, and when they do, we actually see the formation of immune complexes within the gel. Some of these techniques are the ones that we're going to look at first are called passive techniques, because we put the usually the antibody is in the gel, and we load in some patient sample, and we just let it sit. We allow them to react to completion, no extra enhancements. We don't have to apply an electrical charge. And so there are three techniques we're going to look at: radial immunodiffusion, Ochterlony, and electrophoresis. So no enhancements means passive diffusion. There are a couple of factors when we're looking at passive diffusion techniques that we have to consider. Number one is the size of the particles that form. If we end up with very large immune complexes forming, they're not going to travel through the gel, right? The gel is very viscous, and they're not going to move once they form. Temperature can impact it. We want it usually to be at room temperature for optimal testing. The gel viscosity, how was the gel prepared properly? And we have to maintain a hydration of the gel so that it's not drying out. If the gel dries out while we're doing the test, the precipitation is not going to happen properly. And then we're going to look at how well the reactants, the antigen antibody, work within the gel. This isn't a test that works for everyone or for every test. Some of the sources of error in these gel-based tests is if we put in too much reagent or antigen or antibody into the test wells, overfill or underfill them. If you nick the gel with your pipette, you can impact the ability of antibodies or antigens to move through the gel. And also just, you'll see in a minute, the measurement tools are not always super precise, like a ruler. So it's not a perfect system. So the first one that we're going to look at is radial immunodiffusion. Again, this is a passive diffusion method, and it's what we call a single diffusion method. No electrical current is used to speed up the process. And so we're just going to put antigen and antibody there, and we're going to let it rock. The technique is quite simple. We're going to have, when we're making some gel, we're going to add antibody to it. And we're going to mix the gel up, and we're going to put it on a glass slide. So the gel on the glass slide contains agar and a specific antibody that we have in question. Remember, these tests, we're looking for a specific antigen. So we add the antibody that we're hoping that is the specific antibody to the antigen we're hoping to find. So first we put the antibody in the gel, and we slide that all over the whole slide. Then we cut some holes into the agar on the slide. We're going to, put in 4 different holes at least. We are going to put the patient in one of them, and then we're going to use our calibrator standards that we just talked about. So there's a standard 1, standard 2, standard 3. So these have known concentrations of the antigen, right? The antigen is put into these little test wells here. And remember, the antibody is within this gel all over the whole slide. You put it in a little moist chamber and you let it sit and you let it do its job at room temperature. The antigen and the antibody want to meet up. So the antigen starts diffusing through the gel just passively. And when it continues to move, the further it moves away, so here's the point of inoculation, this is all antibody antigen formation of precipitation of immune complexes. So as the antigen leaves and passes through the gel and encounters the appropriate antibody, it forms a complex. And we can visibly see that. It looks like a little hazy ring in the gel. What we're going to do from this is we're going to do a calculation, and we're going to say that the square of so we look at the diameter of this ring of precipitation, and we're going to square it. And that's going to be directly proportional to the antigen concentration. So if standard 1 is level low standard and standard 3 is level high, you can see that standard 3 has a much bigger diameter than standard 1. So the larger the precipitant ring, the larger the concentration of antigen. And so we're going to read it against a standard curve. So if we had a concentration of antigen and the diameter of the precipitant ring that's been squared, so we're going to get a ruler. I'm going to measure from here to here, and then we're going to square it. So if it was, 2 millimeters, we'd square it, so it would be 4 millimeters, and that's what we would count here as the diameter. They have clearly, so standard 1 had a diameter squared of, I don't know, it looks like probably 7, and we know it had a concentration of, again, this is a guess, maybe 6. So it had a concentration of 6 milligrams per deciliter of antigen that we put in the test well. And we read the precipitant ring, and we put a dot. We did the same thing for 2 and 3. We drew the best fit line. Then we took our patient unknown and we measured again the diameter of the ring, squared it, and we got a value of, let's see, probably, I have to go backwards to see it, but let's say we got a standard of 13. I would take it. We did it squared, squared times diameter squared is 13. We take it 13, we go across until we hit the standard curve, then go down, and we can read that we had roughly 11 milligrams per deciliter of antigen in the patient sample. It's a standard curve. Again, look at I'm going to send you the link, and I'll make sure it's uploaded on the PowerPoint that gets uploaded for this, with the correct link. I apologize. And you can see the formation of precipitation within a gel. That's the first technique. So that's radial immunodiffusion. Radial because it goes out in a radius around the point of inoculation, right? The next one is the one that always kills people. It's called Auchterlonie diffusion. And Auchterlonie diffusion is what we call a double diffusion technique. Wells get cut into gel, and we're going to put antibody there, and we're going to put antigen in there. And again, we're going to look for a place where precipitation forms, immune complexes form. And when it does, we see that precipitation within the gel. There are 3 distinct patterns that can be developed in an Auchterlonie diffusion technique that are going to tell us an awful lot about the patient's antigen status. So let's take a look. So in this case, we have a known antibody. We know what this is. Let's do a simple one. Don't look at this one yet. So we buy a reagent antibody for whatever it is we're looking for, and we put it in there. Then we put maybe a patient sample here with an unknown antigen. And then we also put the antigen that's going to directly match with that antibody. And it's going to tell us, in this case, or it's actually going to be it can be a similar antigen, but this is going to be a multi-specific antibody, and this is going to be the patient, and this is going to be a reagent antigen as well. And so what we're going to do is we're going to again put this into a moist chamber, and we're going to let the precipitation take place overnight. We're going to develop lines of precipitation, and you can see them here. And what this is telling us is that if the line of precipitation is sort of equidistant between the patient and this reagent antigen, and it creates this arc like this, this is telling me these are both the same antigen. So if we know what this one was, it was from a reagent, then we know what this one is, because this is the exact same as this. We call this serologic identity or antigenic identity. We call that identity right there. The second thing that happens is, so here's the patient, here's our multi-specific antibody, lots of stuff there, and here's something that we have that's a known antigen. If we get this line that looks like an X through it, and you see how part of it goes, this is called the spur. If you get an X like this, like X marks the spot, right in between the two wells of patient and reagent antigen, this means they are not the same. So if we know what this is, then we know this is not the same thing, and we need to keep looking. So this is what we call non-identity. And finally, the third thing we can get is what we call partial identity. So here again could be the patient, and this is another known, and this one is, you see, we use different reagent antigens here. And this is, these are not exactly alike. We have that spur, and it's facing our known antigen. It's not exactly that arc of identity, but it's not exactly non-identity either. When we get this almost like a T shape, we call that partial identity. And what that's telling us is that these antigens are very closely related. They're cross-reacting, but they're not identical to each other. So we call them partial identity. Partial identity really, again, is just telling us that they're closely related antigens, not exactly the same. So if we look at Ochterlony technique, believe it or not, we still do use this. It's used to identify things like fungal antigens, such as Aspergillus, Blastomyces, Coccidioides, and Candida species. And it's actually put in use still. So, question 2 for our homework. So if you remember, we had question 0, question 1. This is question 2. What does this pattern of Ochterlony tell us about this? So in this case, this is our antibody, and in this case, they actually have 3 wells here. We have a well here and a well here. So this is the patient sample, and this is we'll say this is the patient sample, and this is our antigen 1. What does this shape tell us between the patient and that antigen? What do we know about their relative identity to each other? So you should put that on your paper and let me know what you think. Oops. Electrophoresis is the next technique we're going to look at. This is also a process where we're going to use a couple of different methods with electrophoresis. So the immunodiffusion technique that we just saw, the passive immunodiffusion technique, can be combined with electrical current to kind of speed things along. Electrophoresis is that technique. We spoke about it briefly last week when we were speaking about separation of proteins and immunoglobulins, and we talked about how there is a circuit, electric circuit between within the gel, and we apply that electric circuit to the gel. It's going to help the molecules move a little faster. And just as we saw in our electrophoresis last when we were talking about antibodies, small molecules move faster than big ones. Think of it like a big molecule trying to chug through Jello, right? Small molecules are going to get through it faster. And for immunoelectrophoresis, antigens and antibodies, this is going to help move that faster. So we can do this either by single or double diffusion techniques. There are a couple of different electrophoretic techniques that we're going to look at. Rocket electrophoresis, immunoelectrophoresis, and then immunofixation electrophoresis. First, let's look at the rocket immunophoresis, immunoelectrophoresis. In this case, antigen, again, we have an agarose gel. So we have a gel in a plate, and we've added what to it? Antibody. We've added a known antibody to our gel. That's what we're looking to see if the patient has. So we have these test wells, again, that we cut into the gel. And in this case, this is where we're going to insert patient samples, or maybe we need to put in some standards. We're going to, pipette in a little bit of our antigen source into each one of those wells. And then we apply it into, we're going to apply an electrical current to it. And the antigen migrates in and precipitates it moves through the gel, and where it comes in contact with antibody, you get this kind of an arc shape. And that's exactly what it looks like. The further it moves, it means there's more antigen concentration. So something that doesn't move from the, if you never saw an arc at all, then the antigen doesn't match the antibody. If you see an arc that stays very, very close to the point of inoculation, this is a low one. This one moved very far away, so there was lots of antigen. Remember, we're using up the antigen as it moves along, and when we run out of antigen, that antigen antibody has to be in equivalency. When we run out of antigen, it stops moving. So that would be a high concentration. We can measure the movement, read it off a standard graph, a standard curve, and be able to determine what the patient's antigen concentration was for the control. Electrophoresis, immunoelectrophoresis, is done in a 2D kind of a step, two-dimensional electrophoresis. Antigens first get separated, and this is what we saw last week. When we did antibodies. This is basically the technique that we saw. Antigens get separated first on the basis of electrophoretic mobility. So we had antigen inserted into the well. Antiserum is then added to a trough outside of it, and the antigen moves and the antibody move through the gel together. And where they meet, we get these arcs. We incubate that overnight. Diffusion occurs, and we're going to see that arc at the end of time. So it's a very simple process. It's not anything overwhelming. As I said in the last, so that was immunoelectrophoresis, and I think you can see here. There's the anode and the cathode. So in the test method, we actually have applied an electrical charge to help it move along. And there it is. There we see the electrical charge indicating that it's electrophoresis. So the last reaction that we're talking about for electrophoresis right now is the immunofixation electrophoresis. This process is one that really has been kind of the standard now. Most of the other techniques are not in use at the same level as immunofixation electrophoresis is now. Very much like, the immunoelectrophoresis we just saw, we put unknown antigen into a test well and pherese it through the gel that contains antibody, and that's been applied to the gel. Precipitation forms at the point of antigen-antibody complexes, and there we go. So before applying the antibody, the difference is that let's see if I have a picture of this. I do. In immunofixation electrophoresis, first we add the antigen to 6 lanes of gel. So we're going to apply antigen. We're going to apply it up here, actually. 1, 2, 3, 4, 5, 6. And we're going to allow it to separate. We're going to allow the antigen to just characteristically move through the gel. When that's complete, we go back and we lay over specific antibodies to each one of these. So we saw the antigen move from the point of origin through the gel, and then we lay over antibodies, different ones. So this is let's see, each of the wells. Oh my God, what am I thinking of this? This is all the serum proteins. This is gamma chains, alpha chains, mu chains, kappa chains, and lambda chains. So this is antibodies to those immune complex components. And we look and see what happens. And when we see precipitation form, and we actually add a dye to it, when we see precipitation form, we know that the patient has that within their serum. So in this case, we were looking for gamma chains, and so we do an antibody, an anti-gamma or an anti-IgG, and where we found it, we did see it. And so we measure that against kind of the entire group of serum proteins, and we can see that they line right up. So we can see that the only reactions we really had with this patient was the patient had gamma chains, or, so IgG, that have a gamma heavy chain and a lambda light chain. This is sort of our control here. So this is this. We saw no dye forming here. This is a little background noise. There might be a little something there, but not most of it. So again, instead of applying it to a trough as we did before, we apply it across the whole gel in 6 different lanes. And we'll look and see what the reaction is. It doesn't take long. It takes only about an hour because we're using electrophoresis, so it's much faster, and it's very specific. So in the question 3, you can see here in immunofixation electrophoresis, what does the SP stand for? And the picture that I just showed you, we're going to use the picture before this for this. What immunoglobulin type was tested in this slide? So now we're done with precipitation reactions. Let's look at the agglutination reactions. These are something that I think you've seen before. If you've done a little bit of blood banking in intro, then you've done some agglutination. Remember, agglutination techniques are a little bit different. Agglutination techniques are the clumping together of antigens on cells, could be on microorganisms, some particles like latex beads, and we're going to see how they react with antibody. And we know that because this is a particle that's the antigen is attached to, it's going to be visible. We don't have to wait for immune complexes to be we'll see it. We'll see the agglutination. It's clearly a visible reaction. The particles involved in agglutination can be red cells, and if it's a red cell, we call that hemagglutination. If it's bacteria, we call it co-agglutination. We can also use, as I said, inert particles, things like latex or charcoal beads that are coated with antigen. And we're going to do testing with this that are classified as either direct agglutination, passive agglutination, or reverse passive agglutination. And we're going to look at 3 types of agglutination reactions in the test methods. But first, let's look a little more closely at, sort of the agglutination reactions. As I said, agglutination reactions utilize soluble antibody binding to a particle. So the antibody is soluble always, and the particle is not. So the antigen is on a particle. This reaction, of course, can be visualized. The same 2 first steps that we see in precipitation happens. First is, antibody binding to antigen, and that's sensitization. Is sensitization ever visible? N-O. No. This is the initial binding, and what did we remember about that? It's somewhat stable, but it's reversible, right? It's not necessarily the strongest binding ever. So this is still reversible at this point. The second step is the lattice formation, where one antibody binds to epitopes on multiple cells. Oh, this is still that sensitization. Here we go. The second step is the lattice formation. One antibody binds to the epitopes on cells nearby, and that's the lattice formation. That's how we form those large aggregates and large clumps of cells. This forms a stable lattice, and this is what we're going to be able to measure in our test methods. Let's see. When we look at, good, I don't have it here. An important fact of this is that to make these reactions happen, IgM is a much better antibody for agglutination reactions than IgG. IgM is the most efficient class of antibody in this reaction, really for a couple of reasons. Number one, that initial binding is kind of weak, but IgM has those 10 valences, so it has a far reach in being able to bind out to multiple particles. Also, because it's very big, it may be able to overcome that negative charge of 2 red cells that want to repel each other. You got a long reach to be able to cross the chasm between 2 red cells. And finally, in general, we say that IgM is about 700 times more efficient in agglutination reactions than IgG. So it's definitely a better immunoglobulin for this purpose. What are some of the advantages of agglutination reactions? Super easy to carry out. You put a drop of blood, you put a drop of reagent, you mix it in a test tube. Or you put a latex bead that has antigens on it with a drop of patient serum, you get a reaction. They're quite easy to visualize on a without any extra equipment. So no complicated equipment, no spectrophotometer to read optical densities, no electrophoresis, nothing. Can be performed kind of on demand. It's a really simple test, and a lot of prepackaged kits with known quality control already there. However, with the advantages come some of the disadvantages. Agglutination reactions are just qualitative in nature. What do we mean when we say something is qualitative? I just want to put that in red. We're not going to get an actual number. It's just either kind of a yes or no, present or absent. There we go. Sorry. Qualitative reactions, we're just really talking about present or absent. We're not going to be talking about millimoles or grams per deciliter. We're talking about present, absent. But sometimes that's enough, positive or negative. It's kind of like a pregnancy test. It's either positive or negative. Do you really care how positive it is if you're doing a home pregnancy test? No, not really. We can do something called a titer to give us kind of a semi-quantitative. It's increased, it's decreased. We can give a semi-quantitative value. And then we can also, as I said, we can do a couple of different tests: direct and passive agglutination and reverse passive agglutination. Earlier we spoke about IgMs having, that great efficiency in agglutination reaction. But sometimes we do use antibodies of an IgG class. If we do, the IgG is often too small. The molecule is too small to build a bridge between the two particles that makes that latex reaction, that makes that lattice formation. Why? Well, first of all, IgG is a smaller molecule, much smaller. We saw that it has those strange hinges in each of the subclasses. So in many cases, there's restricted flexibility at the hinge region. And both those issues may prohibit that multivalent binding between two particles, antigens on two different cells or particles. As a result, sometimes we have to do some enhancements to help IgG react properly in a test method. Depends on the reaction, and the conditions, but we may add some things like when you're in blood bank, you'll see that we add some proteins, for example, that may help eliminate some of the issues, or we may add a different test method. We may do a slightly different test method. But IgM antibodies, because they're 10, have a diameter of about 35 nanometers. That repelling of red blood cells is probably only about 18 nanometers apart, I think, if I'm remembering properly. So IgM can cross that barrier very carefully. Let's go to the agglutination enhancement that we do, though, with IgG. Some of the enhancement reactions that we can do can overcome some of the obstacles of that sort of small IgG. Remember, IgG is a great one because it's got nice specificity, but it has those problems of being small and being not too flexible. So we may, for example, add an additive to neutralize the charge in those hemagglutination reactions, those ones with red cells. I told you how they carry a slightly negative charge, keeps them apart. You know what? I think it actually keeps them apart at 25 nanometers, not 18. Not that it matters that much, but I think they keep them apart a little bit closer to 20 to 25 nanometers apart. Perfectly fine for IgM with that, that long arm reach of 35 nanometers in diameter, but for IgG, not so much. So we can add some things. We can add some a low ionic strength saline that we call LIS that neutralizes some of the red cell charge, brings them a little closer together, and allows the agglutination reaction to take place. Maybe we can increase some viscosity. We can use a solution in the blood bank called PEG, which has a goal of reducing the water around the cells, again, allowing that gap to narrow a bit. We can do some enzymatic treatment of the antigen. Going we can agitate or centrifuge the mechanically. And in fact, in the blood bank, we do that little shake of the tube. We're going to agitate it a little, and we centrifuge as well. Changing the temperature of where we do the test reactions actually helps as well. We talked earlier about how IgM likes the cold, anywhere from 4 degrees refrigerated all the way up to room temperature or below. IgG likes it nice and warm, body temperature. And then we can make sure the pH is really optimal in the kind of 6.7 to 7.2 range. And then timing. Maybe we let it sit together for a little bit longer. So knowing what we now know about using IgG, IgM, and the different kinds of agglutination reactions we can have, whether it's hemagglutination, co-agglutination, whether we're using particles that are covered with antigens, what are we looking at? We're going to look at those 3 different kinds of agglutination reaction techniques in the lab. The first we call direct agglutination. These are the reactions where soluble antibody binds to the antigen naturally on the cell. The antigen that's found on a red cell, for example. The antigens that are found on bacteria. That's naturally occurring. We don't have to do anything. Remember, if we're using blood cells, we call the agglutination hemagglutination. Bacteria is co-agglutination. Using direct agglutination is helpful in that it is we don't have to artificially jam some antigen onto a particle. Some other examples of direct agglutination reaction where honestly we just mix, a reagent with the patient sample, whatever the patient sample is. So if we're looking to see if the patient has an antibody, we use a known antigen. If we're looking to see if they have the antigen, we use a known antibody. One known, one unknown, and we figure out if the patient has it. Some diseases like tularemia and rickettsia, typhoid fever, very hard to grow in culture. So we check to see if the patient developed any antibodies to it. Sometimes, as I said, we can use antigens to test to see if the patient has antibodies in their serum. So if we know what the bacteria is, and we can check and see if the patient has an antibody to it, we can figure out what disease the patient has. There's one more test that we have called the Widal test, WIDAL, and this is a rapid screening test used to see if there's typhoid fever. It's a direct agglutination test, and it uses cross-reacting antigens of Salmonella O and some H. flagellar antigens to do it. It's a cross-reaction, so we don't actually have to use typhoid. The bacterial causative agent of typhoid fever, but we can use some cross-reacting ones to see if the patient has the antibody. Let's quickly spend just a second on direct agglutination. I know you guys did this in intro. So blood grouping is a perfect example of direct agglutination. We call that hemagglutination. And our example here is blood group typing. So we have an anti-A, so we're looking to see if the patient has A antigen on their red cells or B antigen on their red cells. So type A has obviously A antigens, but not B. Type B has B antigens, but not A. Type AB has antigens that are both A and B, and type O has neither A nor B. So you can see when you mix A antigens with reagent antibody, you get clumping. The clumping can look like this in a test tube or like this on a glass slide. This indicates, you see no clumping? This indicates no reaction. So if we were doing this, it's not type A because that's blue. Type A is blue. This could be an RH. But we're going to look for that clumping in there. And if we don't see it, it's a negative test. Co-agglutination reactions, as I said, uses bacterial antigen to check and see if the patient has developed antibody. Again, we're going to use it for diseases that are hard to find. Maybe they're hard to grow. We are going to check and see over time if the patient did develop antibody. We're going to check and see over time to see if their antibody titers are going up or down, and that's going to let us know sort of their progress with the disease. Question 4 is, what's the difference between coagglutinin and hemagglutinin? What's the difference? I don't know. Passive agglutination is sometimes called indirect agglutination. And in this case, passive agglutination tests for particles like red cells, latex beads, gelatin, different kinds of artificial silicates. And they get coated with antigens that are not normally found on their surface. So we force antigens onto their surface to use these as a carrier for the antigens. Synthetic beads and particles provide the most consistency because they're manufactured versus a red cell, which are different sizes, different shapes, different lifespan. So instead, synthetic beads are the kind of the best carrier if we can use it. So in other words, we're going to have a known antigen that we're going to attach to a carrier particle, and then we're going to test it against a patient's serum to see if the antibody is present. Very much like we just saw with coagglutination and hemagglutination, but in this case, in passive agglutination, the antigen is artificially attached to the carrier. Agglutination occurs only if the patient has the antibody present to the antigen. We use this for testing, we'll see later on in the semester, to test for things like rheumatoid factor in patients that have rheumatoid arthritis, group A strep. We're going to look for antibodies to group A strep, antibodies to some viruses, and antibodies to things like hepatitis and HIV. The thing to be careful about, though, is, a number of these kits are used to detect IgM. So there can be kind of non-specific binding that can take place as well, leading to some false positive tests. So it's not a perfect test, but it works for us as a rule. The next agglutination reaction we're looking at is what we call reverse passive agglutination. This is used to detect microbial antigens. And some of the common applications for this might include rapid identification of antigens from some infectious things that we don't really want hanging around in a test kit. Group B strep, staphylococcus, some rotavirus, cryptococcus neoformans. You don't need to. So in this case, we're going to detect soluble antigens in the urine, spinal fluid, and serum, not the actual bacteria. And in this case, what happens is I just heard a beep. The process is instead of attaching antigen to the carrier, we're attaching antibody. That's why we're calling it reverse passive. Passive agglutination was attaching antigens to a particle. Reverse passive attaches antibodies to a particle. Again, agglutination occurs if antigen is present and we can identify it. So just comparing reverse passive and reverse passive agglutination. This is from your textbook. So in this case, let's figure out which is which. We have carrier particles that we mix and attach soluble antigens to them. Oh, look at that. So we have particles that we coated with antigen. We add it to the patient's sample with antibody. And if we get lattice formation, agglutination, it's positive. So this is what? This is passive agglutination. If this is passive agglutination, I think that, spoiler alert, this is going to be reverse passive. So we have antibodies and some carrier particles, and we manufacture it so that we can absorb antibody onto the carrier particles. We look and see if the patient has the antigen. We mix the antibody and the antigen together, and if they cause lattice formation and agglutination, bam, positive test. Agglutination inhibition reactions, I think, is our last topic of this. It's not. I think I'm going to break this now. We're going to finish this at the last. Nope, I'm going to do this just for another minute. Agglutination inhibition reactions are another kind of agglutination reaction. In this case, the reactions are based on a competition between the particle and the soluble antigen for antibody. And what this means is, and it's going to be, I hope there's a picture here. There is. Okay. What's important to remember about inhibition reaction is that it's exactly opposite of what we've just seen. So in these agglutination reactions, when we saw lattice formation, it was a positive test, right? We knew the patient had the antibody or the antigen that we were looking for. In agglutination inhibition reactions, it's a lack of agglutination that's going to indicate a positive screen. We're going to look at that in just one second. It's important to remember this fact. We're going to use this to detect certain kind of viruses, measles, mumps, rubella, herpes, HIV, hepatitis B. But let's look at the actual test methodology. I think if we look at this together, this is going to make sense for you. This illustration is in your book. And in this case, let's look at what's taking place in the test tube. So this is a patient who has the antigen in question. So what we do is we have the patient sample that it turns out we don't know when we start. Does the patient have the antigen? Does the patient not have the antigen? We don't know. We go on the assumption that it's possible. So we have the patient sample with unknown happening. We're going to add a reagent antibody for what we're looking for. Again, it might be for hepatitis B, it might be for measles or mumps. We're looking for the antibody to that specific antigen. And the antigen and antibody bind together, right? The antigen takes up all the binding spots here. If the patient doesn't have the antigen, see how there's no binding taking place with the antibody because there was no antigen there. Tons of free binding capacity with these antibodies. What we then do is we add a manufacturer's, a reagent particle that has the antigen in question. So let's use this example that we're looking for, hepatitis B antigen. We want to know if the patient has hepatitis B antigen. So we add anti-hepatitis B. And if they bind, all the antigen binds off the antibody. Then we add particles that we manually coated with hepatitis B antigen, and we add it to the mixture again. In this case, we can't get to the point, we got sensitization, but we can't get to the point of lattice formation because there's no way, there's no free spots for this antibody to bind to the antigen of these particulate reagents. So in this case, if we see no agglutination, it's a positive test. It's a very strange thing. Let's look and see what happens if the patient doesn't have the antigen. Still unknown. We don't know if that soluble antigen is there or not, but we add the antibody to whatever's in question. And in this case, no immune complex is formed because there was no antigen to start with. We add the reagent antigen-coated particles for, again, the example that I was just giving you was the hepatitis B antigen. And in this case, the antibody had capacity to bind to these particles, these reagent particles, and form an immune complex that was visible. We can see agglutination, so that's a negative test. There was no antigen, but agglutination happened. It's because if it's the competition between the patient antigen and the reagent antigen. If the patient has the antigen that attaches to this specific antibody, it covers up all the binding sites for that antigen. So that when we expose it to more antigen, nothing can happen. There's no way for it to bind off. Does agglutination inhibition reaction make sense to you? I hope so. Let's look at another example. This one is going to be hemagglutination inhibition reactions. In this case, we're looking to see if the patient has antibody in their serum. So we have, again, antibody from the patient or a sample from the patient. question mark. Does the patient have antibody to whatever it is we're looking for or not? I don't know. Maybe a virus. We could be looking for a virus, for example. So we have an antibody. In our first test, we're going to add the viral antigen. And because we do have the antibody, the antibody-antigen reaction takes place. There's nothing here to complex with. There's no particle. And this antigen, there's so much antigen, it binds off all the sites of the antibody. We then add reagent red cells that will also bind this antibody. And look, they can't take place. They can't agglutinate because all of the binding sites of the antibody were blocked by this virus. No agglutination tells us the patient had the virus that was in question. In this case, patient did not have the antibody. We added reagent viral antigen to whatever it was in question. Oh, look, no antigen-antibody binding taking place because there was no antibody to start with. We add the reagent red blood cells. And actually, there are receptors on the red blood cells that the virus can attach to, and we get agglutination. If it's yes, it's a negative test. We're inhibiting, making it so this lattice formation cannot form. So it's an inhibition test. We're looking for the lack of agglutination to tell us someone has a positive test. You have to go through this a couple of times to get it. Go through it step by step, take a look at the pictures, read the book around this topic, and it's going to make sense. But you have to do it step by step by step. Inhibition reactions are the opposite. Remember, if we get agglutination, it's a negative test with an inhibition. Always, always, always. We're going to see this consistently, this concept of inhibition reactions as we go forward. So just like we did for precipitation, we have some comparisons of agglutination reactions, and this gives you the interpretation of the results. So there's agglutination means the presence, lack of agglutination in an inhibition reaction indicates the positive test. So this just simplifies a little bit of what we talked about. I'm going to stop on this last little bit and we'll do this in another topic. I want to end the class pretty much on time. So send me your homework questions that come from this, and I will finish this last little bit in another chapter, and I will see you soon. Thanks. Bye.